Genetics, the future of medicine. Our genes are the blueprints of our bodies. They determine our traits, from eye color to our risk for certain diseases. But did you know our genes also play a crucial role in how we respond to medications? This is the exciting field of pharmacogenetics, where we study how variations in our DNA can affect drug efficacy and side effects. Understanding these genetic differences is paving the way for a future of personalized medicine, where treatments are tailored to each individual's unique genetic makeup. Imagine a world where medications are not one size fits all, but are precisely chosen to maximize benefits and minimize risks based on your genes. Decoding our body's blueprint, understanding drug responses. The Human Genome Project, completed in 2003, was a monumental achievement in science. It gave us the complete sequence of the human genome, our entire genetic code. This breakthrough opened up countless possibilities for understanding human health and disease, including how our genes affect drug responses. By decoding the genomes of various species, scientists have discovered surprising relationships between receptor families, the proteins that drugs interact with to exert their effects. These findings highlight the interconnectedness of life on Earth and provide valuable insights into the evolution of drug targets. For example, studying the genomes of simple organisms like fruit flies has revealed unexpected similarities in drug receptor families with humans. These similarities suggest that the basic mechanisms of drug action have been conserved throughout evolution. By studying these shared pathways, scientists can gain a deeper understanding of how drugs work in humans and potentially develop new therapies for a wide range of diseases. The evolution of receptors. From simple organisms to complex beings, receptor proteins, the gatekeepers of our cells, are crucial for drug action. They bind to specific molecules, triggering a cascade of events within the cell. Over millions of years, these receptor proteins have evolved, leading to the diversity we see today. By studying this evolution, scientists can trace the origins of drug targets and understand how they've adapted to different environments and challenges. This knowledge is crucial for developing new drugs that effectively target specific receptors and minimize unintended side effects. For instance, by comparing the structures of receptor proteins in different species, researchers can identify regions that are critical for drug binding. This information is invaluable for designing drugs that are more selective and potent, leading to more effective treatments with fewer side effects. The field of structural biology has made significant advancements in recent years, allowing scientists to visualize these intricate interactions between drugs and their targets in unprecedented detail. Beyond the gene, how our DNA shapes drug responses. The story of our DNA is more complex than just the genes themselves. Non-coding DNA regions, once considered junk DNA, are now known to play a crucial role in regulating gene expression. These regions act like switches, turning genes on or off and influencing how much of a particular protein is produced. Additionally, the spatial organization of chromosomes within the cell's nucleus can impact gene expression. Genes located close to each other on a chromosome are often regulated together, influencing drug responses. This emerging field of epigenetics explores how environmental factors like diet, stress, and exposure to toxins can alter gene expression without changing the underlying DNA sequence. These epigenetic modifications can have profound effects on our health and how we respond to medications. Understanding these dynamic interactions between our genes and the environment is crucial for developing personalized medicine strategies.